fourth week of the stem cell training program. And for those who joined recently, I'm Gitai Kim, Director of the UCLA Human Stem Cell and the Genome Engineering Center. And our center focusing on the both collaborative research and also core service in iPSC reprogramming and genomic engineering, led by Dr. Hyo-Kyung Cha and Dr. Dennis Atta. And this program is supported by the California Institute for Regenerative Medicine in collaboration with Thermo Fisher Scientific. And as a reminder, participants who attended at least 6 out of 7 lectures will receive a certificate of training completion. And even if you have already missed 2 lectures, you can still qualify by attending the missed session next year. So please continue to attend the lecture. For attendance tracking, please log into the Zoom using your full name. And if you logged in with a partial name or different name, and please write your full name and email address in the chat box. And if you are attending without registration, please refer to the chat box and then follow the instruction to register. And you are welcome to ask a question at any time, either directly or through the chat box. Please participate and then interact with the lecturer and then also other supporters. And today we will discuss Lecture 4, Advanced Feed-Free Human Pluripotent Stem Cell Culture, focusing on the optimization strategy and the more detailed workflow. Now I would like to hand over to Omar and Tom.
Perfect. Thank you, Kitai. Let me share my screen real quick.
Hello. Oh, yeah.
Okay, perfect. Can everybody see my slides okay?
Looks good, oh.
Perfect. Thank you. So we are lecture 4, not almost, a little bit more than halfway through. I'm hoping folks are finding the lectures and the content interesting. And like Kitai said, please try to be as engaged as possible in the chat. Drop any questions in there, come off mute at any time. We're happy to take any questions. So we're going to talk a little bit about feeder-free culture today. And just as a review from the past several lectures, right? For the past 3 lectures, Tom and I have talked to you guys a little bit about, what stem cells really are, with their properties, the fact that they can differentiate into pretty much any cell type in your body, as well as their ability to self-renew, the different sources that we can, have the cells from, whether from embryonic stem cells. We also talked about reprogramming a couple of lectures ago. And then Tom also started giving you a little bit of an idea about the different applications that we can start to employ with pluripotent stem cells, from basic research all the way to cell therapy. In fact, if you guys remember, right, this is the stem cell workflow we described very, very early on basically how we typically move and work with these pluripotent stem cells, starting from the left on somatic culture, right, followed by reprogramming, and eventually getting into how we culture these cells after we reprogram them. We're going to talk eventually about characterization and differentiation in another lecture, but today we're going to focus a little bit more on this part of the workflow, the PSC culture. And so before I get started, maybe I'll take this as an opportunity and I'll try to see if I can actually pop the chat up so I can see it a little bit. Maybe I won't be able to do that. That's okay. Maybe Tom, you can monitor the chat for me. But what do folks think we need to take care of or kind of consider when we're culturing these cells? Feel free to drop it in the chat. And Tom, I don't know if you can scream out or shout out any interesting ones that we get. I'll give it
Absolutely.
maybe 30 seconds or so. And if not, no worries at all, right? This is why we're here. So they're
We have
coming in at
CO2, temperature, sterility,
temperature, CO2,
genetic
temperature.
drift.
Okay, interesting. Good, good. All good considerations for sure, right? Specifically for pluripotent stem cells. So what everybody mentioned here is really good considerations for general cell culture, and it is definitely included. Specifically for PSCs, there are special considerations that we also have to take into account. And this is the reason for this is that these cells, and I think we've mentioned this earlier, they're in vivo, they're a transient population of cells, right? We do not carry pluripotent stem cells into adulthood. These cells eventually differentiate. And so when we're culturing them in the lab, right, we need to keep them in very specific conditions that maintain that state, right? And we're going to talk about those today. So I'm going to give you a brief list of some considerations, and then we're going to talk about why we do some of these things and what the reasoning behind them is, right? So the first thing that kind of comes to mind is daily inspections. And what that means is with stem cell culture, these cells can react and change their state quite quickly, right? Almost overnight as well. And so looking at your cells and getting an idea of what they look like morphologically is something that we do regularly. And because these cells have the ability to change, right? Um, Looking at whether there's any differentiated cells or differentiating populations that are emerging has to be considered, and we have to remove those cells as well. And again, the reason for that is primarily because these differentiated cells will start to secrete factors that will cause their neighboring pluripotent cells to also differentiate too, right? So because of that, if we're trying to maintain a pure pluripotent stem cell culture, we have to remove any differentiated tissues that arises. The third is feeding, right? Traditionally, with pluripotent stem cell media, we had to feed our cells every single day. In fact, this is a running joke between stem cell researchers is that we would consider them our babies because they required so much daily attention. Now, we will talk about this today as well, a little bit more in detail, but more recent or more modern media systems allow for flexible feeding, where you don't have to feed every single day. And these are newer innovations that we're going to talk about today. Um, Ahead of actually, passaging your cells, right, or splitting them, and this is something that Tom covered last time, and we're going to cover again today, right? But as your cells continue to grow in culture, there's only a limited amount of space, right? And so if they use up all that space, then they start to crowd, they start to fight for resources, and again, their pluripotency status might be at risk, right? So, in order to make sure we're ready to split these cells when they become confluent, we have to make sure that we have everything that we've prepped beforehand, right? Before the day that we're supposed to passage or split these cells. And so one thing that we have to keep in mind is all those things that we need to prepare before we even passage as well. Things like preparing your dishes, coating them, preparing meth plates as well, which we're going to talk about a little bit today. And close to that, right? How are you passaging your cells when they get to that, confluent or confluency that requires splitting, right? These cells are very, very sensitive, and so for routine culture, we don't recommend passaging them as single cells, where you dissociate everything that's attached onto your plate, and you dissociate them into single cells, and then move them all over and split them into the next passage. Instead, what we recommend is passaging PSC cultures as small clumps or colonies. This way, they can remain attached to each other to a certain degree, which will help them fare, that stressful splitting journey, if you will, a little bit better. So these are just some of the things that, I think are top of mind that are important to keep in mind. And we're going to talk a little bit more about them. But we talked a lot about stem cell culture, what they are, what they should look like, and or how we use them, excuse me. But what do they look like, right? What should we be looking for, and how do we know if we have a good versus bad culture? Was there a question? Sorry. Maybe somebody was off mute. No worries. But if we look at the first thing, right, that I mentioned before about daily inspections, we primarily look for how these cells, fare in culture by assessing their morphological features. And we look for very specific features in our cultures with pluripotent stem cells that are quite unique compared to other cell culture or culturing other cell types. Mainly is that those cells like to be, very close knit with each other. So they'll grow in colonies as you see over here. And those colonies are very, very distinct in the fact that their cell contact between the cells is quite close. So the colonies should be very compact. They're going to have a nice rounded edge, as you can see over here. In this case, you'll see also what's called the refractive edge, right? It almost looks like somebody's shining a light from behind, right? And it's reflecting off of the edges here. And that's because the surface of the edge of this colony is slightly beveled, right? And that's again another morphological feature that we look for, these smooth, rounded colony edges that are refractive. Um, Now you'll notice some other cell types here in the back, and so these are mouse embryonic fibroblasts that these pluripotent stem cells are cultured on top. These are very, very different, right? These are not pluripotent stem cells here. These are mesenchymal fibroblasts, but they're used in this case as just a feeder layer for these cells to sit on top of, which we'll talk about. But if we zoom in slightly, right, and we look even closer at higher magnification, you'll see, right, within this colony, the really, really close cell-cell contact that I was talking about. But you'll also notice that pluripotent stem cells in general have a very high nucleus to cytoplasm ratio, meaning that the nucleus makes up the bulk of the cell size, if you will. That also said, you'll also notice that within the nucleus, in each cell, there's sometimes multiple prominent nucleoli. That's another also feature of PSC morphology. Shane, you have your hand up. Do you have a question?
Yes. So for somebody who already has a lot of experience culturing hESCs, how much how much is different about iPSCs in your experience? Are many of the concepts around the handling the same, or is are there things that take a little bit longer to learn with iPSCs versus ESCs?
Excellent question. So typically the protocols would be very, very similar, I would say. In certain cases, with certain cell lines, ESCs can even be a little bit more sensitive as well compared to iPSCs. Not always, but generally speaking, that seems to be the trend. But in terms of morphology, they should behave very, very similarly, Shane. So great question. If you already have some human ESC experience, that would be transferable into the iPSC culture as well.
I'd like to ask a short follow-up question.
Please.
For somebody who is transitioning from hESCs to iPSCs, what's the biggest, most important difference in the culturing and the handling?
There isn't going to be much that's different in terms of the handling part. What you're going to see the difference is going to be in terms of the kinetics of the cell line themselves. iPSCs tend to grow slightly faster. Not always, but sometimes. And so their morphology and how they grow might be a little bit different to reflect their growth kinetics as opposed to human ESCs. Um, Now, with that in mind too, there are differences like on a transcriptomic level, for example, Shane. So what I'm mentioning here is it's more about how you take care of the cells. There is slight differences between ESCs and iPSCs. There's certain for methyltransferases, for example, that are different expressions, things like that. But generally speaking, the way you take care of them is very, very similar.
Can I mention one more thing?
Please, yeah, please keep
Yeah,
that.
yeah. Also, in usually when we access ESL, they are wild type. And usually we are not just collecting the mutant version, but iPSL, our more interest is just isolate iPSL from the patient. And then depending on the mutation, their behavior is quite different. And then
That's a good
sometimes
point.
those mutation affect the pluripotency is very, very hard to maintain because they have very strong differentiation potential. Yeah, just a
True.
quick note.
True, true. Absolutely. Absolutely. Great call out too about the difference in terms of knowing a clinical outcome versus not, right? So with ESC chain, that material is derived from early embryo sources, right? So you really don't know what the endpoint is or if there's a disease that might emerge as related to the genetics of the sample that you collected. For iPSCs, because we're able to collect them at a later time and reprogram the somatic cells, you might be able to, depending on the disease that you're studying and the stage at which you collect, right? Either early during development or later on during adulthood, you might be able to say this person has a particular, pathology or disease and use that sample to reprogram, right? And so to Kitai's point, the genetic background will make the differences here. In wild type cases, there shouldn't be much difference, but again, it is really highly dependent from one cell line to the other. I hope that
So
answers your question.
it does. So then, do you think, Dr. Kim, that there's more differentiation potential in iPSCs and that you have to take more care to avoid having them differentiate during your culturing than is the case for HESCs?
Yeah, I totally agree with Omar. At this moment, ESL, iPSL, and the wild type, very hard to see the difference. And but the genetic mutation, not all of the mutations, some mutation, and then those genes typically express in pluripotent stem cell. In that case, we need to be careful because there is some kind of relate function is defective. That is my point.
Yeah. And Shane, one other thing is what I've described you where you saw the like this image, right? This is a really nice colony. And there are some certain iPSC cell lines that once you reprogram them, they won't look 100% like this. They may look a little spread out or something like that. But generally speaking, with a wild type genetic background, they should adopt this morphology as opposed to something that could be slightly different. But it's not out of the blue to see something slightly different on the iPSC side when it comes to different mutational backgrounds or different patient samples that have been reprogrammed. Great
Thank you.
questions. I love this. Sweet. So, well, now that we kind of have an idea of what they should look like, well, what happens if things are bad or if we do not maintain those conditions that, require or that maintain these cells as pluripotent and they start differentiating? Once they do, you can clearly tell from at least 2D monolayer culture, right? Because this is what starts to happen. The cells start to flatten out a little bit more and they start to diffuse out, right? Almost. They're not necessarily migrating, but you could see if you go into closer magnification that the cells are really flattening out as opposed to the image before, and that their cell broad bodies almost spread out a little bit more. And because of that, you might also start to see that this refined, colony edge is no longer there, right? It's that refractive, dome almost is again spread out. And sometimes this will manifest in really spiky cells at the edge of those colonies. And that spiky morphology typically indicates a stress on the cells, right? That the cells are not too happy. They're potentially trying to figure out a way to get out of there. And as a result, they end up differentiating. right? And it's not also uncommon to see, some differentiation on the edges of the colony or in the center, where other areas of that colony might still have a slight resemblance of traditional PSC morphology that we just talked about. But again, in this case, this is a differentiating culture that eventually these cells will impact any pluripotent stem cells that are, in the center here, that will eventually differentiate. And so because of that, right, when we see those differentiating cells in our culture, we recommend removing them, right, or what we call cleanup. And this is honestly as easy as taking a pipette and a sterile pipette or a syringe and scraping off those cells under a microscope so that they're no longer attached before you change the media to remove them, right? And one thing you'll see here again, kind of explained, is that when these cells differentiate, the cells don't always look kind of or don't always adopt one morphology. So you may have this flattened kind of morphology that still remains next to the colony, but it is dispersed out. You might have these satellite cells that are kind of a little bit off those edges. You might have pockets of these flattened cells inside of a colony, right? So they might look a little bit, they might present themselves differently, but they're very, very distinct, right? And when we see them, oops, excuse me, did I go? Yeah, once we see them, we kind of like, this animation is kind of showing. We take a sterile tip or a pipette and we scrape them off. And so when we do, right, we try to get as many of those differentiated cells off as possible so that they are not influencing the remaining pluripotent stem cells as well. Okay, so we kind of have an idea now about what the cells should look like, what they would look like if they start differentiating, and what we might be able to do there. But let's talk about how we maintain them in this really good kind of state, right? In a state that will preserve their pluripotent status, right? And I think Tom showed you this, or a version of this depiction last lecture of basically four things that we have to consider when we're looking at human PSC culture. This is, not specifically related to, or these four things aren't just specifically related to pluripotent stem cells. In fact, you can adopt this kind of format in thinking about any cell culture. But we're going to talk about specifically about what matrix, what media and what passaging considerations we have to take specifically with pluripotent stem cells. So let's start with matrix, right? I've mentioned before, and you might have heard Tom also mention this, is that pluripotent stem cells have been traditionally very hard to culture, right? In the first introductory lecture where we talked about how old our field is, right? Going back to the 1880s or the 1950s, depending on how you look at it, and the limitations really of the field advancing being in how we could harness the power of these cells by culturing them in vitro, right? And traditionally, these cells did not like to sit on plastic whatsoever. So when the original work came out, and as it was transferred into human culture as well, pluripotent stem cells were grown on top of a layer of mouse embryonic fibroblasts, or MEFs, and sometimes they'll be called feeders as well. And so what these cells did is that these cells would attach to the plastic, and then the pluripotent stem cells would attach on top of them, while the MEFs also provided some support to these growing pluripotent stem cells, right? For example, in mouse PSC culture, these mouse embryonic fibroblasts secrete LIF, which is a growth factor that's really, really important to maintain the pluripotency of mouse PSCs. Now, uh, when transitioning over into human culture, right, this was a little bit more problematic because We had now mouse embryonic fibroblasts sitting underneath human pluripotent stem cells. So any downstream assays that we do, we had to either parse through the two different cell types or be content with having, two different cells that we're analyzing at the same time. But as you all know, with science, we like to isolate variables where if we want to make a deterministic call, right, or try to establish a, cause like a cause and effect, right? We have to just manipulate one factor, right? And so because of that, we moved away, or we're starting to move away from mass cultures in human PSC side, and we've adopted a more feeder-free format. But the properties of stem cells remain, right? They do not like to sit on plastic. So what do we do there, right? In this case, we can coat the plastic with various different, matrices or growth factors in order to get them to stick. But we can't just use anything. There's specific ones that have been validated that we should use. The first is laminins, right? Pluripotent stem cells really like laminins. It's an environment that they are familiar with in vivo, right? This is what they see a lot of laminins in the growing embryo. So attaching to a laminin-based matrix, whether laminin 521 or laminin 511 as well, is really, really favorable. These cells really like it and they stick really well to that substrate. In fact, when we're doing, very harsh workflows with these cells, which we'll talk about in a little bit, like gene editing or single cell sorting, where we know these cells might be stressed during that process, we use a laminin-based matrix to ensure better attachment of these cells. Another matrix that we can use to coat the plastic is vitronectin. There is a specific N-truncated form that we'll talk about of vitronectin that is used in order to coat the plastics, but this is a kind of a xeno-free offering that also has a pathway to the clinic because there are clinically compliant versions of vitronectin, like the CTS vitronectin that I've also included here. But more commonly, you'll see basal membrane extracts being used. Things like Geltrex Flex or Matrigel are used in order to coat the plastics before the pluripotent stem cells are seated on top. And basal membrane extracts are kind of a really good alternative here for the specific reason is that basal membrane extracts is basically taking the extracellular proteins from the basal membrane. In this case, of a mouse cancer or sarcoma, and because this extracellular matrix has so many different proteins that include laminins, integrins, fibronectins, collagens, it's a good surface that might be good for multiple cell types. And in this case, it is as well good for pluripotent stem cells that can be used. So again, here, the distinction of whether you're using MAFs, right, or mouse embryonic fibroblasts, that you see here on the on the left, excuse me, or matrices that you see on the right will dictate whether your culture is feeder dependent or feeder free, right? And as I mentioned, I don't like to think that one method is better than the other. I think one method may be more suitable, right, or appropriate depending on what your end goals are, right? So in this case, for human PSC culture with with a goal of going to the clinic, feeder-free cultures provide that environment that allows you to isolate variables maybe a little bit better in this case. And with that kept in mind, right, I just as a reference for you, I've made this table of these three different matrix or substrates that you can coat the plastic, a short description of what they are, whether they're xeno-free or not. And what xeno-free really means is that do they contain any animal origin components that might, cause them to be might cause the matrix to be at risk when you're going to those clinical endpoints, right, or going towards the clinic. And relatively speaking, they come at different costs as well. So just keep that in mind. Another thing to kind of keep in mind is that although the different substrates are compatible with with PSC culture, the way in which they're prepared is a little bit different, right? And so their thaw temperatures might be a little bit different. Their storage and stability might be a little bit different. The recommended coating, as well as the dilutants, right? What you're diluting, the stock that you're buying before you coat it onto the plate, right? So again, this is just as a reference for you for everybody, but please keep these things in mind as you're preparing your plates, right? This is really, really important because if your plates aren't coated well, the cells are not going to attach. So you're not even going to be able to see what the cells look like under the microscope the day or 2 days later. Any questions about matrices so far? Maybe in the chat, but we'll see. We can. So let's move on and talk about the second thing, which is maybe arguably the one of the more important aspects of PSC culture is media conditions and supplements, right? What are you providing to those cells in order to maintain that pluripotent stem cell state? And over the years, this has been innovated on quite a bit, right? Tom and I like to talk a lot about this of how far we've come along in the field. Where in the late 90s, right, we were still in the using feeders, but in terms of media, we were using basal media like DMEM or DMEM F12 and supplementing it with as much as 20% FBS in order to get the cells to grow a little bit. And these were ESC cultures. And even then, you really had to pay very close attention and work really, really intricately with these cells because that system was very, very error prone. And if one thing, happened, it could mean the demise of the cultures. Eventually, right, not too far in, or the early 2000s, we started replacing FBS with knockout serum replacement as a way to maybe control a little bit more the supplements that we're adding. It was still not defined, just like FBS is not defined, but KSR or knockout serum replacement is a bit more defined than FBS, so it helped reduce some of that variability that's associated from different lot to lot that we see with FBS. Eventually, right in 2006, 2007, a group in Madison, Wisconsin, actually came up with what they called defined feeder-free media, which was M-teaser, and they were using that with Matrigel, which is a basement membrane extract, in order to establish feeder-free cultures for the first time, right? Which was revolutionary in 2006, 2007. But since then, that same group also, right, established Essential 8 media, which is basically a pared down version of a PSC media that only contains the bare essentials, right? 8 essential components, as the name suggests, in order to, again, try to control downstream variability a little bit more. At that time, right, we were still trying to understand how to culture these cells properly, what they should look like, any deviations that they might have, that might be caused if we change culture parameters. And in this case, Jamie Thompson's group really wanted to make sure that they have a xeno-free, fully defined media that they can again potentially take into further downstream applications. And then since then, I can speak at least on Thermo's behalf. We've been trying to innovate as well in this space where we've launched different versions of the Essential 8 media that help with daily feeding, as we'll talk about in a little bit, as well as different medias like StemFlex media that are more geared towards modern applications like gene editing, single cell sorting. Again, we'll talk about that in just a second, but more support for these cells as we use them in different ways with modern workflows, right? So let's break this down a little bit further, right? Let's start with the earliest onset with feeder-dependent cultures, right? I mentioned that originally we were using FBS supplemented media, right? We're adding anywhere between 10 to 20% FBS. And so KSR came as a replacement, a direct replacement for FBS. that would help control, growth and maintenance a little bit better, help with PSC cryopreservation, and actually has also been published and shown to be more effective in terms of at least mouse ESC line derivations, right? As opposed to using FBS. So in this case, the feeder-dependent media would be your DMEM F12, which is your basal media, KSR, your knockout serum replacement, which is intended to replace the FBS, as well as some standard, supplement or cell culture supplements like non-essential amino acids, mercaptoethanol, and then in this case, FGF, which is again a prerequisite for mouse ESC culture, right? And in this case, transitioning from FBS supplemented to KSR supplemented media is quite simple. It's almost like a one-to-one replacement, and you really see those really nice dome-shaped PSC cultures. These are mouse PSC cultures that you're seeing here. Now, on the human side, right, like I mentioned, we're moving towards more feeder free systems, right? And so this is where more modern media is favored. And for feeder free applications, the way I like to bucket, the way I think about PSC media is almost twofold, right? And kind of described here. Leaner media that you see on the left, like Essential 8, it's as the name suggests, lean, meaning it contains, and I hate describing it this way because it is a good media, but it contains the minimum requirements that these cells need to maintain that pluripotent status, right? We know what, there's been work done in this case to figure out what are the components that are needed to maintain these cells as pluripotent stem cells. On the right side, or kind of like the other side of that coin, there's these more robust, more modern PSC media systems. And at Thermo Fisher, we have our Stemplex media, for example, that is designed for more modern applications, right? It provides a little bit more support to the cells and allows you to use these cells in more modern workflows like gene editing, single cell sorting, that we can employ with these cells, right? And we'll talk more about that too. But if we're talking about Essential 8, right? Essential 8 media was derived from Jamie Thompson's lab, which is the same lab that described, the first defined system, right, with mTeaser. And again, in this case, the goal here was to reduce variability by reducing the number of components that you need to add into your formulation, right? And the logic there was if I need to, if there's less ingredients that I have to make, right, that I need to make the media, then there'll be less variability every time I make it, right? If you take this analogy, for example, if you have to cook something, and cooking something with 2 or 3 ingredients tends to be a little bit simpler than cooking something with, 50 ingredients, right? And so this was the case. And in this effort, Jamie actually, in a Nature paper, Jamie's group described how they've, come to testing the different components and what is really needed for these cells to maintain this really nice PSC colony morphology that we described earlier. Now, with the Essential 8, actually in the same paper, Jamie's group also started talking about what matrices, right? This is when we started shifting away from feeder cultures and into feeder-free cultures, right? And here they tested coating plastics with vitronectin, which is an extracellular matrix protein, right? And they tested, coating the plastics with multiple different variants of this protein, the wild type variant, a truncated C terminus, a truncated N terminus version, and then another version that had both C and N termini truncated. And as you can see here, the N truncated version seemed to support the attachment of these cells, the best. So together with Essential 8, Jamie's group really described the first serum-free, xeno-free culture media systems for human PSC culture, which was really, really exciting, right? Now, one of those components that Jamie identified, right, as being essential in PSC maintenance and PSC culture in vitro is FGF, right? And it's actually part of the ingredients, if you will, or the components that you add to an essential eight media. But FGF is quite special in this case, and we should talk a little bit more about it because FGF is not heat stable, meaning at 37 degrees, right, which is the temperature that I think people called out, right? When I asked that question of what parameter should we consider, somebody called that temperature, right? So this is where we culture our cells in the incubator at 37 degrees. But at 37 degrees, the FGF degrades quite quickly. And so you can see in older media systems like Essential 8 or MTeaser 1, there's a huge drop in FGF concentrations in that first, 24 hours when you're culturing the media. And again, this is just media without cells as well, but at 37 degrees, right? And you can see actually if at time zero, for example, here, less than 10 hours later, there's less than, half of that, of the original FGF in your media now. Now, this was a little bit problematic, right? And the way we got around it at the time was we figured, well, okay, there's this huge drop in FGF concentrations, right, overnight, and we knew that this is a very critical component that we had to keep in supplementing to our cells. So what we did was we actually fed our cells every single day, right? So these older media systems, like Essential 8 or MTeaser 1, they require daily feeding, right? And this is the reason for that, again, is to resupplement and bring up the FGF concentrations again. Now, with more modern systems, you see, right, like StemFlex, Essential 8 Flex, MTeaser Plus, for example, the FGF concentrations are maintained a little bit better in the formulations there due to more recent innovation. And so I will say that this was pioneered at Thermo Fisher, right, with Essential 8 Flex. So this was originally done with that Essential 8 media, but stabilizing the FGF2 activity for up to 72 hours, and eventually transferred into our StemFlex formulations as well for more robust support for these cells. But again, the goal here is when you stabilize FGF2 concentration is in addition to, again, reducing variability, right? You don't want these FGF2 levels to keep fluctuating up and down within that span of 24 hours up until you feed. And again, what this really allows us to do, right, in addition to reducing variability there, is not feed every day, right? We can passage our cells and then feed them on, the day after. We can keep them incubating and let them grow without disturbing them or changing too much of their environment. And when they're ready, we can passage them. And there are options with StemFlex and Essential 8 Flex where you could, for example, on a Friday, if your cultures aren't too confluent and you know that they can still grow, there's space for them to grow for another day or two, you can do what's called a double feed and add twice as much media if you're using those flexible feeding systems and incubate it over the entire weekend without having to replace media there, right? Now, this for traditional PSC or PSC researchers, if you had said that to us maybe 15 years ago, people would have gasped, right? What do you mean? I leave my cells the entire weekend without coming in, changing media, looking at the cells, making sure everything's okay. But this is how far we've come, actually. And we have a lot of data to show that we can, use these flexible feeding systems and flexible feeding schedules. We can maintain pluripotency. If we do that in these older systems, we cannot. And we can actually also use those flexible feeding systems for long-term culture. Here I'm showing an example where we've maintained cultures for up to 60 passages in StemFlex medium. And you could see that even after a very long time in vitro culture. I don't know if I would recommend people work with really, really high passage numbers with PSCs because they can potentially accumulate mutations right in culture. But at least with the flexible feeding systems, the morphology looks really good. We've also karyotyped these at passage 52, and they're karyotypically normal, and they maintain normal pluripotency or stemness markers. So again, the using the right media system for you is really important, right? But how do you make this decision? And it's a bit challenging, right? And so this is a good kind of reference point that you can use in terms of how do you figure out what works best for you, right? The way, again, I like to think about it is, do I want to be in feeder dependent or feeder free culture? Am I doing some basic research or going to more translational routes? And based on that, you can assess different kind of parameters that would help you discern. In this case, for example, if I'm looking towards a gene editing workflow, right? I'm going to make a point mutation in a cell line to mimic some sort of disease, very similar to what Tom described last lecture, right? With generating those hypoimmune iPSCs or iPSC cell line. I might want to use a media similar to StemFlex. That would be, the best support for my cells during those gene editing workflows. If I'm concerned about knowing exactly what's in my formulation, right? And I want to know every single component because I want to know how it interacts with certain pathways, I might be thinking about Essential 8 or Essential 8 Flex because I know every single component in that media formulation, right? So just a couple of things to think about. Now with StemFlex, right, and more modern media that are more robust, right? One really added advantage, like I mentioned, is the ability to work in those newer workflows, right? Gene editing, single cell sorting, right? And so I told you guys that pluripotent stem cells do not like to be singularized, or they do not like to be dissociated to a point where they're just single cells. They will usually differentiate. And but that can be a little bit tricky, right? When you're trying to do gene editing, but the cells don't really like it when they're on their own. So how do you figure out a way where you can edit the cells and then screen which cells has the right edit and only grow that clone, right? Where you do clonal expansion. And so in traditional media systems, when we do this, right, when we do single cell cloning, where we see the single cell in a single well of a 96 well plate, for example, and we see what conditions would allow the single cell to grow back into a full colony, right? It might be a little bit stressed out, but can we prevent it from dying or differentiating and allow it to grow back into a colony that we can screen for our desired edit, right? And so if we do that with Essential 8 media on a Geltrex matrix, about 0 to 5% of our of these single cells that we've seeded will actually result in a clone that we can then work with, right? So very, very low. We can alter this a little bit by adding different supplements like ROCK inhibitors, like the Revitacell supplement. And then I mentioned to you guys, laminin matrices are very supportive for the attachment of these cells, right? So when we do that, we change conditions, still plating these cells, these single cells in essentially media, we see a slight increase in clonal efficiency, where about 15% or so of those individual single cells can grow back into a colony that we can propagate and screen. But where we see the biggest difference is when you add, again, an appropriately designed media for these applications like StemFlex, where this is a robust media, again, that's intended for supporting those very rigorous, harsh workflows. And there we see the cloning efficiency shoot up to 45%, right? And the reason why this is important, right, is especially in gene editing workflows, is if you don't pick the right media for these workflows, right, you might end up repeating experiments over and over again in the hopes that you'll get, one colony or two within that 5% range, right? Whereas with more optimized conditions, more optimized media systems, you do have the luxury of, hopefully picking from different clones and choosing which ones that you want to work with specifically, as opposed to just by chance or by luck. I won't go into this into too much detail as well, but just for your reference, gene editing is an up and coming workflow that we're routinely now using with pluripotent stem cells. But so is single cell passaging, right? Where potentially for when we're differentiating, you might want to have to count cells and you have to seed them or plate them down to a certain concentration. And so single cell passaging is also another consideration for these more robust media systems Again, there's multiple out there, but I can only speak to Thermo Fisher's StemFlex at this point. Perfect. And then the last thing we'll talk about in terms of the considerations for PSC culture in general is passaging, right? And Tom, you might have seen this slide or remember this slide from Tom's lecture on what passaging is, right? So passaging, as most of you will already know, is just subculturing of the cells where once they've reached a certain confluency, meaning that they've occupied enough space within your culture vessel, it's recommended that we lift them off the plate and then split them and into different vessel formats so that they're not competing for space and resources, right? And for pluripotent stem cells, this is really, really important, right? Because again, these cells, if they're stressed, if they don't have the right nutrients or growth factors, they will differentiate. So maintaining them below a 70% or 80% confluency is really, really important. And specifically the passaging method, right? How we lift them off and how we move them over into the next culture vessel is also something that should be paid close attention to, right? With ESC and iPSC passaging, the dissociation reagent that you use will impact how quickly the cells and colonies are kind of separated or dissociated. And so there are different dissociation reagents that are commonly used that range from collagenase or dispase that are used traditionally in early mouse and feeder dependent cultures. Triple and acutase are a little bit stronger so that they can dissociate your colony into single cells or sometimes into small clusters as well, depending on how long you incubate them for. But traditionally, with routine PSC culture, a EDTA-based association methods are what's going to give you that clump passaging that we talked about in at the beginning of our lecture, right? Where you're not passaging the cells as single cells, you're more passaging them as clumps. So let's see if this works, and I hope this works. I'll turn this off. Let me know if you could see this, but I wanted to show you what that looks like in practice, right? So these are three different dissociation reagents. I'll pause it here. On the left, you have Versine, which is an EDTA-based reagent. This is going to be a gentler reagent. In the middle, you have Acutase, again, a stronger dissociation reagent. Some can place it in that middle ground. And then on the right, you have Triple Select, which is an alternative to trypsin that we commonly use as well for cell culture, can get you down to that single cell suspension. So let's see how these three different reagents, kind of how they fare with our stem cell culture. Starting with Versine, right? So Versine is, like I said, EDTA as an EDTA-based solution. It's a non-enzymatic digestion where you're basically you're using chelating ions to promote cell dissociation. And you can see that here in this front. But what results in happening is that you have these clumps that then you can passage over into your next culture. And these clumps sit really nicely, right? They will attach, and then in 24 hours, that's what they look like, right? They're going to start to adopt that PSC morphology. Now, if we redo this work, right, but instead we use StemPro Acutase, which is a proteolytic enzyme-based dissociation reagent, right? It's a little bit stronger. In this case, you can see those, 1 to 2 cell clusters. The dissociation is happening a little bit faster, and you could see the cells kind of coming apart, right? And once you passage them over or move them over into your next vessel, you'll see single cells, but you'll also see 1 to 2 cell clusters as well, right? That can be counted, but again, it's not a 100% full single cell dissociation. And in this case, you'll see the cells do attach, but they have a slightly more spindle-like morphology in the first 24 hours as they're acclimating, because it is a stressful event to bring them down to that level of dissociation, right? And so if we move over into Triple Select, which is the stronger out of the three reagents, that gets us down to that single cell passaging or single cell suspension, you'll see this very, very clearly, right? Compared to the other two. You're really going down to a single cell dissociation here, where I can almost count this cells as they're dissociating, right? And if we move them over, if we move these cells over into our next passage or our next culture vessel, this is what they'll look like, a single cell, right? And just remember here that in this case, I am reseeding the cells into stem flex medium, right? And stem flex medium was designed for this specific application, but you'll notice that the morphology of the cells is very spindly. They look quite stressed, right? This is normal for that first 24 hours once you do single cell passaging, because it's not something that these cells are accustomed to, and it's not something that they like either. Perfect. And so, like I mentioned, for routine culture, we typically recommend using EDTA-based dissociation reagents, like Versine, for example, in order to get clumps. But again, you have to pay attention to how long you leave your dissociation reagent incubating with your cells. And a good reference here is sort of what a good clump size versus small clump size would look like. Because if your clumps are, too small, the cells are still going to be stressed because they're not going to have that cell-cell contact that they prefer. If they're too big, then they might fold in on themselves as they're, being plated back down into your next passage, and might differentiate because you're creating necrotic core because of the way the clump folded in on itself. So a good sized clump is typically achieved with a good wash in around 3 to 4 minutes of dissociation with EDTA or Versine at 37 degrees. But it can change between different cell lines, right? So figuring out what works best for the cell line that you're using again is again very, very important. There we go. I I've okay, let's see. I'm not gonna play this entire video, but what I also wanted to just show is there's a lot of resources online that we can help with. I put a link here to another protocol on how to cryopreserve the cells. And you know what? Actually, we do have time, so let's run this through. When you're dissociating your PSC culture, right? You can either dissociate them in order to passage it over into the next culture vessel to continue propagating your cell line, in your incubators, or you can opt to potentially cryopreserve these cells that you've dissociated, right? This is a very short how-to video, again, linked here, and there's a little bit more of those that I can point to, which will show as as needed throughout the lecture series. But this is a really good resource for you to have on what we necessarily do to dissociate and cryopreserve ourselves as well. Like I said, I won't go through the whole thing, but as you can see here, the first thing that we do is that we wash our cultures to make sure that we're removing any media or any reagents that could block our dissociation, right? We then incubate, in this case with Versine solution at 37 degrees for 4 to 5 minutes. Speed this along pretty quickly. And once that's done, the really nice thing about these using these chelating agents is that once you incubate them, the cells are still very loosely attached to your culture vessel, right? So what you see me doing here is I've actually aspirated the media first, which you would think would aspirate all the cells, but since they're loosely attached, they are still remaining on the plastic a little bit. And in this case, after I remove the Versine or the EDTA, I'm going in with a little bit more media to wash any cells that are loosely attached that, again, I can keep in those clumps and cryopreserve them as such, right? So you see me here just washing the culture dish so I can get any of the loosely attached cells. After that, we're going to take them over and spin them down. We're going to put them in a tube, spin those down. See that? Usually we spin for around 4 minutes at 400 Gs, after which you should be able to see a very small cell pellet that you see here at the bottom, right? Right over here. After that, we're going to very carefully aspirate that spent media, making sure that we're not touching the cell pellet. And then we're going to add in our cryopreservation media quite slowly. Just make sure that your cryopreservation media is kept on ice, uh, and that you're adding it slowly to the cells. Notice how I'm adding first dropwise and I'm rocking the tube back and forth. These are little tricks that you'll learn with watching some of those videos. And the reason why I'm doing that is I'm trying to reduce the osmotic pressure that the cells are feeling as they're experiencing the cold cryopreservation media as well, right? After that, I'm going to invert to mix my cells very, very gently. So I don't want to froth or pipette up and down too vigorously. And then move over my cells now in the cryopreservation media into a cryo vial. There we go. And again, notice how I'm adding in dropwise first before I, completely dispense the media out. These are little tricks, again, that will help reduce osmotic pressure, reduce stress on the cells as you're handling them. And eventually now that that cryo vial can be, put in a Mr. Frosty, stored at -80 for overnight before being moved to liquid nitrogen the next day as well. Sweet. Any questions about the cryopreservation? Whoops, I keep moving forward. Tom, any questions in the chat?
No, just going through them, Omar. Some great ones, but
Some?
yeah, we're all good.
Awesome. Perfect. So let's recap, right? We talked about daily inspections, right? And the reason for that is, these cells, even though I've shown you some really, really beautiful cultures and beautiful images, sometimes, things don't go as planned. Sometimes they differentiate. So making sure that you're, aware of their morphology, what they look like, and then removing anything that does not look like stem cells is important. How you feed them is important and what you're feeding them is important, right? So which media system are you using? Is it more of the traditional media systems where you have to feed every single day? Or is it more of the modern media systems where you can employ flexible feeding schedules or potentially even modern applications where you have to consider media as also as a confounding factor that might affect the efficiency of your experiment or your success of your experiment? Preparing, plates and reagents ahead of time, right? For example, when you're passaging your cells, you already need to have coated your next vessel, right? Prior to dissociating the cells and moving them over. And that coating process takes, anywhere between an hour to 2 hours. So you have to, prep those things ahead in advance. And as we just discussed, when you're passaging, at least for routine culture or routine maintenance, it's really important to reduce the stress that these cells are experiencing. And an easy, simple way to do that during routine culture is to make sure that you're passaging them as clumps and not as single cells or cluster or small clusters either. So everything I mentioned was, talking about how we culture these cells in monolayer or in 2 dimensions, right? But in more recent years, we've also started, and many of you have already heard this, about how organoids and 3D biology is really, really taking a mainstream, right? Really trying to reflect, maybe selfishly, but our 3D biology as well, right? And how that changes how we think about the way the cells behave in culture. So, over the past several years, there's been attempts to culture these cells in 3D or as spheroids. And so at Thermo, we do have a media system that allows you to do that called stem scale, where the idea is you form these free-floating aggregates of these cells that maintain pluripotency. So we're not forming embryoid bodies. We'll talk about that when we talk about differentiation. But we're keeping these pluripotent stem cells, but just aggregated in spheroid form, and they're being cultured in what we call suspension culture. So they're not attached to anything. They're just a spheroid floating around, right? In these cultures that are being agitated. And the nice thing about these suspension-based systems, right, similar like stem scale, for example, is the fact that now that you don't have to worry about the surface area or the area available for these cells to grow, right? We can now play with the volume of the media in order to scale up or to get more cells or culture more cells at one given point. And this is a very important consideration when we're talking about clinical applications, right? Where we might need billions of these cells at a time in order to use them as repair kits, right? So culturing them in 2D might be a little bit harder because you're basically stuck with surfaces, or you're stuck to the surface area constraint of your culture vessel. Now, this is not a new kind of methodology, if you will, about cell culture in general, right? Even for pluripotent stem cells, there have been folks that looked at ways in which they can potentially use microcarrier beads in order to attach the pluripotent stem cells on, or doing spheroid culture. But um, previously or traditionally, right, we had a very hard time implementing this because microcarrier cultures were, they did provide consistent growth, but the attachment of the cells to these microcarriers was not very consistent. And if we're going to use these for any clinical applications, the microbeads themselves had to be removed before, final use, right? So that also carried some translational risk. Spheroid cultures, again, were also implemented. They were relatively easy to maintain at the time, but what we originally had a problem with as a field in general is how do we maintain the center of these spheroids and keep it as pluripotent, right? Making sure that it's not necrotic, that all of these nutrients and gases are able to penetrate to the center of the spheroid. And and so with more modern, suspension-based PSC systems specifically, right, we were able to maintain the expansion capabilities of these suspension media systems, simplify workflows, and make sure that we can scale across different formats. But very importantly, right, to make sure that we're keeping the pluripotency status of the cells is maintain consistent size of these spheroids as they're growing. So we're able to deal with them again more consistently throughout the culture period. And if you think about this, right, if you think about, well, if I have cells in a 6-well plate that I want to grow and get billions of cells for a certain experiment. And in this slide, you'll see that the ask here is to create 4.5 billion cells, which is roughly the number of cells that we need to treat 2 patients with cardiomyocyte cell therapy, right? If I'm starting with 1 well of a 6-well plate, which usually, yields around 1 to 1.5 million cells, if I'm going through traditional adherent culture or monolayer culture that we've been talking about, I will start with these 6-well plates, have to go to dishes, eventually go to cell factories, and getting that 4.5 billion cells will take about 28 days, roughly so. If we scale in these suspension-based workflows, we get that same cell number, but in a shorter period of time. We end up using less media as well, so it's less expensive as a process, which again is more amendable for these translational endpoints, right? Where cost and time is also a consideration that we have. But let's talk about these suspension-based systems, right? What do we need? What can we do? Again, there's multiple different systems that are out there. I'm going to speak to the system that we have at Thermo Fisher, but just know that, like Kitai said on previous calls, that there are other options out there. I can't speak to any of them specifically, but I'll let you know what our systems can do and what considerations you should pay attention to. Sweet. So starting off with our 2D culture, right? We can take our 2D culture that you see over here and then dissociate it with our stem-pro-acutase. And if you remember, the stem-pro-acutase dissociation can get us down to these, single cell or, 1 to 2 cell clusters as well, right? And so when we do that, we then replate these cells, but in our stem scale media, right? So this is a media that was specifically designed for suspension-based culture, right? I see some folks are drawing on this stuff, which is absolutely fine. Please go ahead and draw on my slides. Um, um, Now, uh, once you replate these cells, you have to make sure that you're replating them in specific culture vessels. And so what do I mean by that? You see these two plates, or this plate and this flask over here. I'm calling them out specifically because they have to be non-TC traded or low attachment plates because you don't want any cells attaching to the sides of the wall in suspension-based workflows. This is not specific to stem cells. You'll see these types of plates being used, in any 3D, settings. But an important consideration compared to traditional 2D culture plates that are treated for cell culture specifically. And so when we see these cells in those non-TC treated plates and stem scale media, we also add a ROCK inhibitor. And in this case, we're calling out the Y27632 compound as our ROCK inhibitor here. And the reason why we add this ROCK inhibitor is to get these cells to reorganize their cytoskeleton a little bit to a degree where they extend out some projections, causing an increase in the surface area of the cell itself, right? And so this is a neat little trick that allows you to improve the way these cells nucleate. Because we have dissociated with the stem-pro-acutase, we're down to a single cell, almost a single cell suspension, right? And so now we have to reform those single cells into these aggregates or these spheroids, right? And the addition of the ROCK inhibitor allows you to do that, right? Without its addition, these cells won't be able to stick to each other as easily and form those spheroids. So let's take a look at how this looks like with this process of initiation. Well, is it playing? Okay, it is playing. Again, there is multiple videos here that talk about maintenance and passaging of these cultures. I won't go through all of them. I'll mention the processes briefly, but I won't show the videos. But they're all there for you to see, and I'm happy to share any and all of it. First thing you see me do here, which honestly we should have covered a little bit, but make sure your workstation is clean. And what I start doing is I'm prepping my plates, right? For 2D culture, we talked about coating. For 3D culture, we don't have to coat. But what I do is, you see here, I'm adding my stem scale media into the culture vessel that I'm going to be seeding my cells into. Okay. So this is what you need in order to initiate your stem scale cultures, right? You need the dissociation reagent. You need some buffer for washing that has no calcium, no magnesium, right? Typically DPBS. The complete media, which is our basal media of the supplement, and then your ROCK inhibitor and non-TC treated plates. So the first thing you do is you take your 2D culture, like I mentioned before, you aspirate your media, and you wash these cultures with DPBS, right? With no calcium, no magnesium. The reason why we're washing is that we're prepping for us to dissociate these cells, right? So we have to make sure we remove any remnants of the media so that will block our dissociation. Once the cells are washed with the DPBS, you'll see me add the stem-pro-acutase to start the dissociation process and place the cells in the incubator for 4 to 5 minutes. Once those are done, you'll see that I can collect my cells from now they should be in around single cell or 1 to 2 cell clusters. and I'm going to add that cell suspension to a little bit more media so I can stop that further dissociation of the cells, right? We don't want to keep the cells in a solution that will continue to dissociate them further. So after that, we spin the cells down, and then we'll take the cell pellet and resuspend it in our stem scale media with that ROCK inhibitor that I mentioned. And then if you notice, and I'll pause it here. If you notice, this plate was the original plate I had prepared at the very, very beginning with just the media that I put into my incubator. So I took that one out of the incubator, and now I'm using my cell suspension here with the stem scale media and the ROCK inhibitor and adding the recommended 100,000 to 150,000 cells per ml. And you see here, I've actually done it in plates as well as in a shaker flask. And the final thing is with suspension-based system, you have to make sure that your cells are constantly agitated. Otherwise, the cells will just settle to the bottom, they will clump, and then they will differentiate. So in this case, putting it on an orbital shaker with a 19 millimeter throw, setting it on a 70 rounds per minute or RPM, will give you some really, really nice consistent spheroids. That should have it on the next head, right? That will form in less than 24 hours. I see, by the way, it's the same marking here. Is there a way to remove it? Kim, Kita, I'm not sure if, or is that me actually?
Omar, I've been trying to remove it and I cannot, unfortunately. It might be on your end if you go to annotate under the three dots.
Let me see if I could just remove it. There is.
Sorry.
Aha, there we go. Got it.
I mean,
No worries.
I knew you were bad at drawing, but I didn't know you were that bad at drawing.
I'm terrible at drawing. Sorry, everybody. Sorry about that. Okay, so once you seed those single cells, just the way I showed you, right? It's a pretty simple process, doesn't take too much. But in less than 18 to 24 hours, those single cells, because of the ROCK inhibitor as well that you added, will rearrange their cytoskeleton and become a little bit more sticky. And as they're being, moved around in that orbital shaker, they're going to find each other, and then they're going to undergo a process called nucleation, where they kind of, start binding to each other and forming those spheroids, right? And on day one, they're visible. You can kind of see them a little bit. But at this day, we recommend feeding the cultures, which means that you simply change the media. And we'll talk about the media changes for these systems here. This stem scale media also has a flexible feeding technology, so we don't have to feed our cells every single day. FGF is stabilized in this formulation, so we can skip feeding on day 2. And around day 3 to day 4, you'll start to see the cells or the spheroids that have nucleated have grown big enough to the point where we now have to passage them, meaning that we have to dissociate them again, back down into single cells, and reform them because there's a lot more cells now compared to day 0. In fact, typically we see around a 5 to 10x expansion in those 4 days, 3 to 4 days, in a single passage with this media system. And so this is what these cultures look like, right? These are 4 representative images, 2 iPS cell lines and 2 ES cell lines. And you could see here that the size of the spheroids is relatively homogeneous when it comes to their diameter, as well as their morphology, right? Which is a really important consideration for suspension-based systems, right? This is super, super important because if you have a system where you do have spheroids growing in culture, but some are big and some are too small, when will you know when to feed them or when to passage them? How will you know that the center of the big core of the big spheroid is not, starting to experience hypoxic conditions or necrotic cores compared to a smaller one, right? So having a relatively homogeneous culture is a lot easier to deal with. And in this case, as I mentioned before, from day 0 onwards, as these spheroids nucleate and continue to grow, their diameter increases. And we use their diameter in this case as a benchmark for when we should passage suspension-based systems, right? Typically, I recommend passaging when the cells get to around 400, when the spheroids get to around 400 microns in size, because closer to the 500 range is when we start to worry about, the limits of diffusion, right? And then you might start to see necrotic cores, potentially cell death in the center of that spheroid that might not be exposed to as much air, as much nutrients, oxygen, nitrogen, compared to the outer periphery of the spheroids. Now, within that time, right, feeding those cultures is important, right? So a single passage in this suspension-based system is around, like I said, 3 to 4 days. But we typically feed the cells at least once or twice, right, during that period. So how do we do that when there's cells floating around and they're not attached, right, compared to traditional 2D systems? And this is quite simple as well. If you're working in plates, all you have to do is remove your plate off of agitation from the orbital shaker in the incubator, and then prop it up at around a 45-degree angle up against a rack. There's even these really cool racks now that you can buy specifically for plates. And just wait for 5 minutes, where the spheroids are going to now settle to the bottom center of that dish, right? And then from that point on, you can just with the pipette aspirate only half of the spent media and replace it with a fresh half, right? If you're working with shaker flasks, the process is even simpler. You don't have to tilt it up against anything. You just remove the flask from the incubator. You set it down for 5 minutes for the spheroids to settle to the bottom. And then you aspirate 50% of the spent media carefully and replace it with 50% fresh media. Now, I did mention that the ROCK inhibitor was really essential when you're initiating those cultures. But very much like 2D culture, we do not recommend constant addition of ROCK inhibitor during feeding, right? Because our goal, again, is not to stress these cells out more than we have to. We use a ROCK inhibitor in 2D in case for better attachment and in 3D for better nucleation. But once we're feeding, right, the cells, we don't need to supplement with ROCK inhibitor anymore. But you'll also notice that I've mentioned that you only need to do 50% medium replacement, right? This is not something that is, general to all media systems or suspension media systems. This is specific to the way this formulation is designed. But again, this is maybe another call out for when picking your culture media system in general, or the way you want to model your workflows. Make sure that you take into account, the different platforms that you're employing. In this case, right, one of the advantages of using stem scale medium is that one, we don't have to feed every other day, or we don't have to feed every day. We can feed every other day. But when we do feed, we only have to do a 50% medium replacement. And that is by design, right? So this is the way the media was designed. And again, when you're looking at your workflow constraints, make sure that your process is simple. If you're working towards, clinical scales, make sure your process is amendable to automation. And with suspension-based systems, it's very, very important to look at maintaining, low shear force, because the media is moving around. It's almost like setting your cells up in what is it called? A wave pool, right? The ones where the waves crash, right? If they're in there for long enough and the waves are strong enough, it might damage your cells, right? So in these suspension-based systems, it's again important to make sure that you're maintaining constant shear force, and accumulating and preventing the accumulation of wastes as well. Now, when it comes to passaging these cells, again, the process is quite simple. All you have to do is remove your culture vessel from agitation from the incubator, collect your spheroids in a conical tube, spin those down, And then the resulting, pellet, you're going to aspirate spent media and resuspend that pellet in stem proacutase so that you can read, digest, or read dissociate those spheroids back down into single cells. And once you have them as single cells, you're going to do the exact same thing we talked about in the beginning on how to initiate those cultures. You're going to take that single cell suspension, resuspend those single cells in stem scale media with a ROCK inhibitor, and then seed them in your new culture vessel. We talked a little bit about this, but again, in terms of passaging considerations, make sure that your process or you're picking up platforms that are, easy when it comes to, the manipulation part, that you're minimizing handling time so that you can minimize contamination risks and cell loss. And again, with these suspension-based systems, because of their nature and the way they're used for clinical applications, making sure that you do have at least a pathway to scale and automation can be something to think about as well. Now, I did mention that with stem scale, right, and suspension-based media systems in general, right, the goal is to get, a large cell number usually, right? And this is true here where we do see that 5 to 10x expansion every passage, at least with the stem scale media system. And as folks scale up, and you'll see this often, a lot of the work that we initially do, we don't want to do in very large formats, right? We want to do in smaller scale, make sure that we have proof of concept work before we start, scaling up work and generating millions or billions of cells that we can use for clinical applications. And so making sure if you're working in suspension-based systems that, again, you have a pathway to scale up, right? Making sure that you are able to go from smaller size flasks to larger size flasks or 6-well plates to bioreactors, making sure you maintain the identity of the cells and your expansion potential is really, really important. And as you can see here, for stem scale, this is exactly what happens. We can scale up our cultures from these small flasks to 1 liter flasks, maintaining the same 5 to 10x expansion. We can also do the same if we're going from plates all the way to bioreactors. In fact, if anybody is looking for a little bit more support there or knows of anybody looking for support, we have refined protocols for suspension-based systems up to 3-liter vessels and now working on even higher, where, in one passage in those 3-liter vessels, we can get 5 billion cells at a time, which is extremely, extremely cool. Now, Tom touched on this slightly last lecture again, and we will talk more about this, but a really, really important consideration, especially if you're moving from a traditional 2D system to a 3D system that you have not used before, is to make sure that the platforms that you're using are maintaining the identity of the cells as pluripotent stem cells, right? And so for this suspension-based system, what we did in order to, again, make sure that the media is not affecting long-term culture or providing a selective pressure on the cells where we're selecting for only mutated cells, for example. We've passaged these suspension-based culture systems for 30 consecutive passages, right? So we form the spheroids, we let them grow, we then dissociate the spheroid, then reform it, and we do that process 30 times. And after 30 passages, we looked at, pluripotency markers like OCT4 and NANOG, making sure that they're highly expressed. We looked at global gene expressions and validated them against what we traditionally know to be traditional transcriptomic signatures of even 2D culture. And we also looked at, karyotypic, if there's any karyotypic abnormalities and any point mutations in proto-oncogenes. And thankfully, none of that was present. So we were able to use the system, or we're very comfortable, using the system for downstream applications or long-term culture as well. Now, I won't, spoil the fun and ruin the differentiation lecture that's coming up in a couple of weeks. But I do want to give us a small teaser on as folks start to move into also 3D systems as well, we're starting to learn about how we can start to differentiate those 3D systems completely in suspension, right? And I've given just an example here where we have a thermo a differentiation kit that allows you to differentiate your pluripotent stem cells into dopaminergic neurons specifically. And this kit, as you can see over here, was developed originally for 2D monolayer culture, where you do a 10-day initial induction into a floor plate progenitor state. You then expand these progenitors for about 10 days, and then you start maturing them into dopaminergic neurons. And what we did after we derived or we were able to kind of finalize the stem scale medium, the stem scale system is now use those PSC spheroids as our starting point for our differentiation, and really playing around with the differentiation protocol parameters and in order to see whether we can achieve the same thing and get maybe midbrain organoids instead of just a flat 2D layer of dopaminergic neurons. And as you can see here, we're able to get these really cool progenitors. Again, I won't spoil the fun, but we're able to get this and going and even have systems that we can plop onto multi-electrode assays that can physically, fire and cause bursts to occur. And this we can do this not just for neuronal subtypes. Again, these PSC spheroids that you see here in panel A, grown in stem scale, can be taken down other differentiation protocols. You could see in this case, in this example, we're differentiating into mesoderm cardiac organoids. And in about 14 weeks, you could see these really cool beating organoids that like spontaneously beating culture and almost, they look cystic a little bit. There could be some potential chambers. We're in the process of characterizing them right now and looking at the different cell types that emerge. Really, really, really cool work that we're going to talk about in the next several lectures too. But I'll stop there for the sake of time. And then I just added some, helpful resources. There is a bunch of free e-learning courses that you can take online around, thawing PSCs in 2D systems like in StemFlex or how to culture your cells in suspension with stem scale. I've also linked a whole bunch of other how-to videos over here that are really, really cool. All ranging from like media selection to media preparation to considerations during culture, gene editing talks, things like that. Yeah, and with that, I'm happy to take any questions. Any questions the group may have? Let me see if I
I
can
think we got
stop.
most of them in the chat, Omar, but there were some really good questions that came in start to finish on
Nice.
your talk. So well done.
Awesome. I'm glad to hear it. Let me see if I can stop sharing so I can see you guys again. Okay. Do I see you again? No. I don't know why, but I feel like I'm becoming an old man. Okay, I think I did this. No. Am I sharing anything on your end? Ah, there we go. Stop sharing. Oh, done. Awesome. T Tom, any interesting questions you want to call out
Yeah,
to
we
the
had
group?
some really good questions around basically culture conditions, culturing under. Well, actually, one really good question that came in was culturing PSCs at 5% CO2, right,
Hmm.
or 20% O2 versus hypoxic conditions. I thought that was
Excellent.
really interesting. Obviously, different situations when each of those are needed. But there were some other ones on matrices as well.
Yes.
Yeah, some really good ones, honestly, today.
Awesome. Speaking of hypoxia, actually, this is a good thing. Just as a little side note, PSCs are somewhat resilient in 2D to hypoxic conditions because in a very weird way that's almost normoxic for them, right? Early conditions in the developing embryo can be a little bit hypoxic, so these cells are used to it. In fact, I think I mentioned this in the reprogramming lecture. We can increase reprogramming efficiency by culturing the cells in hypoxic conditions from either day 3 or day 7, depending on which cell type that you're reprogramming. But even when you rederive these cells, you can grow them in hypoxic conditions. I've gone down to 1% O2 as well, and it is tolerated. And I will say this, that's not to say that, you could just throw in these cells in hypoxic conditions and just let them be. There are certain considerations, and you have to make sure that you're doing this for the right reason specifically. Um, I see a couple hands up. I might have sparked some interest. Uh, Noreen, you want to go first?
Sure, thanks, Omar. And so I have a question about quality control testing. Are you going to cover any quality control testing in the next lectures, or?
You mean in terms of like characterizing the cells or quality control for the reagents and the media?
No, characterizing the cells and, after that, like reprogramming or.
It's true. We will cover that. I think Tom, is that in the next lecture before we get to the differentiation, right?
Yeah, I believe so.
Yes. Yeah, no, we will cover that for sure. It's a
Okay.
very, very important consideration because as you already know, if you're not starting with the right population of cells, your downstream endpoint is going to be different. And if you think about it, it's almost like a ripple effect with these cells. A small change in upstream can really make a difference once you take the cells down and you differentiate them into a host of progenitors before a terminal cell type, right? So very, very good call out. And it's actually something that the field is being a little bit more focused on, generally speaking. We have recommendations now from the International Society of Stem Cell Research that we try to follow as well. So we'll talk about that for sure.
Thanks.
Great question. Shane, you had your hand up as well? You had a question?
Yes, so I am I'm guessing these cells are relying primarily on glycolysis for their ATP. I think that's generally the case with stem cells, and based on the fact you say that
Do
you
you
can take
please
the oxygen
the
the oxygen
presentation
levels
requested
down
for the long
to
report
very
notes for
low.
postdocs?
Is that what we're
Rather than
dealing with?
6 months old.
Okay.
There seems to be somebody off mute. Oh, there we go. Okay. Sorry about that, Shane. Go ahead.
So these cells are relying primarily on glycolysis for their energy. Is that right, for their ATP?
Uh, I would say yes, they can. That's the thing. It's a shift, right? That they can have, but yes, they can rely primarily on glycolysis, I would say.
Thank you.
But yeah, the reason why I didn't want to say that, Shane, is like the extended period, right? It's how much can they tolerate? Because past a certain point, right, then they will start to stress out, and then they will differentiate, right? So it's like a matter of exposure, but they can tolerate it. Excellent. Great questions. I loved all the engagements. I'm just scrolling through the chat. This is amazing. Um, Sweet. So I hope this wasn't too boring, and I hope it was informative. If anybody has any questions even after the call, please let me or Tom know. We're definitely happy to help. But Kitai, I'll pass it to you for closing remarks.
Yeah. Tom, any remark?
No, that was great. Thank you, Omar. I love the kind
Okay.
of take it one step further on the feeder-free culture. So really good. Thanks.
Yeah, thank you, Omar and Tom, for excellent lecture. As discussed, while some examples are based on the Thermo Fisher product and the underlying principle, composition, and the methodologies are broadly applicable to similar system and the platform. Thus, even if the media and the material differ from what you are currently using, it will still be one of the similar system introduced by Omar. So these concepts are highly relevant for improving robustness and the reproducibility in human pluripotent stem cell. For the next week, we will move into exciting lecture, gene editing in human pluripotent stem cell. And this session will be extended from cell culture to genome engineering strategy and the application in stem cell system. Thank you again for participation, and we look forward to seeing you next week. Okay, bye.
Thanks, everybody.
Thank
Have a good
you.
one.